So we will first talk about the definition of clinical fungi, because yesterday we have already introduced what are fungi, how do they live, and how do they disperse. And so we can skip this part. And then we talk about antifungals, and at the end we discuss briefly some classification of fungal diseases, which are called mycosis. So, and in the mycosis, in the third part, we will also introduce some very special cases of diseases caused by fungi in humans, for introducing some principles in the scientific treatment of fungal diseases. So when we talk about fungi which can infect humans, we have the principal problem how to confirm that fungus is causing a disease in humans, and how to prove that scientifically. Because when a person is getting sick, when it's getting ill, then there can be many different reasons, and so it's very important to find the real reason. And in science, in particularly in pathology, we rely on the so-called Koch's postulates. So you see here is a painting of Mr. Koch. Robert Koch lived in the 19th century to the beginning of the 20th century, and he elaborated the scientific principles for defining pathogens. So we have a look on these so-called Koch's postulates. So we first describe it in words, and then I will also try to explain this in a figure. So when we have a disease, and then we should find a specific microorganism which is present in this disease. And then we should isolate this microorganism and use this microorganism in an infection experiment in a healthy organism, and this should lead to a new disease in the originally healthy organism. And then this is a third point. This is not yet enough, because we should try to find this same microorganism in the newly infected and diseased organism again. And this should be the same as the initial one. So, and by these 4 steps, all these postulates are fulfilled. If there is one step missing, then it's scientifically not very appropriate to talk about the disease agent. So again, this is shown in a graph, the same principle. At the beginning, we have a diseased organism, and then we detect and isolate the microorganism associated with this disease. And then we transfer this isolated microorganism to a healthy organism, and then check whether the healthy organism is becoming diseased. And there should be the same symptoms as in the originally diseased organism. The symptoms look like the same. And then we should isolate again the microorganism and check whether it is the same as the originally isolated organism, in order to be sure that this is the same. So this is a very logical structure and scientifically very clear. And when these postulates are fulfilled, then we have the scientific proof that microorganism is causing a disease. But in humans, of course, we cannot do infection experiment so easy. So not so easily. So in animals and in plants, the Koch's postulates are very frequently applied, uh, and but in humans you cannot do such infection experiments because of ethical reasons, right? So we cannot just use the students for doing the infection experiments for us, professors, and then we note how many students get ill by our infection. That is impossible. So in human diseases, just in the most important for us ourselves, the most important diseases, we cannot really apply these scientific postulates. So we need other criteria. So, and here I want to share with you because in the very beginning, when we started a collaboration with a physician from a hospital and wanted to submit our first paper about a new fungus associated with a human disease. And then we got the feedback from the reviewers. And this feedback, we couldn't find it in any publication. But this was very important because we cannot do infection experiments. So we need other criteria for defining so-called clinical fungi. This means fungi which can cause diseases in humans. And so the reviewer shared with us their criteria, and now I'm sharing them with you. So the first is a fungus already known as a clinical fungus? If this is the case, if I detect fungus, for example, from my fingernails, and I identify it, and then I found in the literature many records of this fungus from fingernails, and perhaps only from humans, then it's very likely that my new finding is correct, right? Then the reader will accept my finding. But when I find a fungus which has never been recorded from humans before, but perhaps from plants or from soil, then the reader will be very critical and will ask for more proof that this fungus is really causing the disease. So I hope you can understand this principle. So when we compare our new findings with findings from previous people, and then we should see the degree of novelty. So if we only add some additional data to already existing knowledge, that is okay, but it's not a breakthrough, but it's acceptable for the majority of people. But if we find something completely new, which has never been recorded by anyone before, then we need much stricter evidence for this finding. So then the second question from the reviewers was this fungus which we have isolated the only organism isolated? Because in the human body, there are usually many different kinds of microorganisms. You already know that there can be bacteria, fungi, and viruses, and other microorganisms. So the question was only referring to fungi, but actually, if we want to be sure, we should check also perhaps other microorganisms. So if I isolate the particular fungus among many other fungi and bacteria, then the evidence is not so strong. But if I only found this single species in several isolates from the same patient, from the same person, but no any other organism, then the evidence is better. So then the third question is also in the same direction. On how many separate occasions was the organism cultured from the same patient? So that is also referring to the time. So because the diseased person can visit the doctor several times, and at every time we can try to make isolates of the microorganisms. And when the disease symptoms are always present and we always isolate the same microorganism, then that's also quite good evidence. So in the fourth question is just in the same sampling. When I take samples from the same person in the same time, I can take several samples, and how much is the percentage of the fungus from this number of samples. So if, for example, I have 5 samples and isolate the fungus only once, that is not so strong as I isolate the fungus from all 5 samples. And of course, when we take several samples over the time, and then the evidence will be also more important. So the fifth point is also very important in clinical fungi, in clinical mycology, and because we can directly observe the fungus in the human tissue. Yesterday I have shown to you that fungi can be present as hyphae and as spores. So, and both kinds of cells can also be directly observed in the human samples. So when you find such structures and can show them with photographs directly, this is a very strong evidence. If you have, when you only believe or tell you believe that there is a fungus, or you have some indirect methods, then the evidence is weaker. So, and the last one is referring to the treatment. When you treat the disease with medicaments, perhaps specific against fungi. So we will talk about such antifungal medicaments later. So if you treat the symptoms with medicaments, and then will the fungus disappear so that it cannot be found or isolated again anymore? If the fungus is disappearing, this means that the medicament was successful, and so this is also a kind of evidence which is accepted for defining clinical fungi. So when you just can eradicate the fungus with your treatment. So, and because we cannot apply the scientific course postulates, so we need some other kinds of evidence, and we just have an accumulation of evidences. And the more evidences we get, the better. But we cannot get 100% perfect evidence as with the course postulates. So in any case, our criteria are not as good as the course postulates, but they are still better than having no evidence at all. So that is a reason why I'm sharing with you these criteria for defining clinical fungi. So now we have a look on the infection of humans by fungi. Yesterday I told to you that fungal spores are very widespread in the air, about 200 spores to 200,000 spores per cubic meter. So, and the spores are all present in the air. So we are inhaling, we are breathing in fungal spores every time. And then the spores enter our lung, our body. And so the human body is already co-evolved with the presence of fungi. So when the humankind was coming up during evolution, there was already the interaction with the fungi. So our human body is already well adapted against the infection by fungi. So always there's a contact, but humans are actually rarely infected by fungi. So, for example, the first point is the human body has high temperature, so at least 35 or 36 degree. And so many fungi cannot grow well at this high temperature. So that is already one reason why many animals and humans have such high body temperature, because this is already very effective in preventing infection. So only fungi which can survive and grow at higher temperature than 35 degree, they can potentially infect humans and grow in the human body. That is the first condition for a fungus for becoming infective. And the second is the ability to overcome the immune reactions, because the human body has some other methods, not only the high temperature, for preventing infections. I think you may have heard about that in other lectures. And I will show that also in the next slide. And then the third point is in the human body, there are also some specific compounds which are rare in the environment. And so these compounds are not very frequently encountered by microorganisms, and therefore general microorganisms from the environment do not have the ability to degrade these compounds. And the most important one is keratin. So the surface of our skin, our nails, our hairs is all composed of keratin. But this is very outside the human realm and outside the animals. Keratin is very rare. So keratin, for example, does not occur in the plants. So fungi which usually grow on plants or in the soil do not interact with keratin. So only if they are able to degrade keratin, then they have the potential to infect humans. So here we have three conditions which must be met or should be met before a fungus could be infective to humans, before a fungus from the environment could become a clinical fungus and cause disease in humans. So here's an example of the immune response in the human blood against infection by fungi. You know, in the blood we have the white blood cells, and the white blood cells behave like amoeba. And so if they come in contact with some foreign organisms like bacteria or like fungal cells, in this case we have yeast, the yeast Candida albicans. And then the white blood cell will recognize, oh, this does not belong to the human body. And then it will enclose this foreign object, and then the foreign object becomes enclosed in a vacuole. And then inside the vacuole, the microbe is digested, and then it's disappearing. So that is an example, a very simple example of the immune response. And so only microorganisms which can avoid or can counteract this kind of immune response, for example, by blood cells, such organisms only can become infective and can cause diseases. So, and then that is a third point which I mentioned, that there's a specific keratin. So the upper surface of the skin and the nails and the hair is composed of keratin, and this is very hardly degradable. It is a protein which can very hardly be degraded because there are very strong disulfide bonds, SS bonds. Because there is an amino acid in this protein is very common, which is called cysteine, and this has these SS bonds, the disulfide bonds, and these are very strong bonds, and so the keratin, if it is degraded by microbes, it's usually not only degraded by specific enzymes, but also by the reduction of these sulfide bonds with sulfide directly. So that is also an aspect. But when the keratin is degraded, it is split into its amino acids, so the cysteine is released, and when the cysteine itself is degraded, then the sulfide inside the cysteine is also released. And both cysteine itself and sulfide are generally toxic for cells. So when you, including fungal cells, so when a fungus is able to degrade keratin, then the cysteine and the sulfide are released and become toxic to the fungus itself. So this is causing a great problem for the fungus, which must also be solved for the fungus. So keratinolytic fungi, these are fungi which can degrade, which can lyse keratin, and they can solve this problem not only by dissolving keratin, but also by degradation of cysteine and by exporting the sulfide outside the cell. So that is a first step in the keratin decomposition. There are specific enzymes which we call keratinases, and then they can degrade the disulfide bond in the keratin. And then the keratin becomes separated into small fragments. So yesterday I have shown to you that the fungal life is depending on such way, the fungal feeding. It is excreting enzymes into the environment in large quantities. And then by chance the enzymes encounter the substrate, and then they degrade the substrate. And at the end we have some very small components, very small fragments, which can be taken up again by the fungal cell, and then inside the fungal cell they are digested. So that is an example. Actually, we do not have only a single enzyme, but different enzymes which interact and help each other in the degradation of keratin. So on the left side we see a model of the keratin molecule. We have, for example, filamentous structure on the left side, and the filament also composed of amino acids on the right side. So the one blue bubble may represent an amino acid, and the amino acids among each other are also connected time by time by the disulfide bonds, and these are very difficult to break down. So in the first instance, in order to separate these two filaments, we have the first is here, the yellow symbol is an enzyme which can split these bonds, and then the left line and the right line with the blue dots can already be separated. And then the other enzymes can access these molecules from all sides more easily when the two filaments are separated. So we see this here. Here's a green symbol, which is just cutting off the amino acid from one end. And on the other side, we also have the same enzyme also cutting the keratin filament from one end. So, and at the same time, we have other kinds of enzymes. These are the violet symbols here, which can cut the protein inside the molecule. So, and by these processes, we get smaller and smaller fragments of the keratin protein, and at the end we have very short fragments, and then there is again another enzyme which can finally split the remaining amino acids from these very small fragments. So we need not to memorize all these names and so on and all these steps, but just understand that the degradation of the keratin is a very complex process. So it's not so easy to evolve all these enzymes specifically for degrading keratin. So in this graph, we want to have another focus. So on the right side, with a brick-like symbols, like a wall, we have the keratin. So this illustration is a symbol that the keratin is like a very strong wall, which cannot be broken so easily. And on the left side, we have the outline of the right side of the fungal cell. So here the whole thing should be the cell of the fungus which can degrade keratin. So and what we see first is that the fungus is exporting sulfide for splitting the disulfide bonds. On the one hand, there is the enzyme, a specific enzyme is involved, but this enzyme needs the assistance of sulfide for splitting these bonds. And then there are the different kinds of enzymes, the other kinds of enzymes, which continue to split the protein into the amino acids. And then yesterday we have learned when the substrate is split into its small components, and then the components will diffuse, and some particles come in contact with the plasma membrane of the fungus, which is indicated here on the left side. And these fragments, now these are the amino acids from the keratin. They come in contact by chance with the plasma membrane, and then a vesicle is produced, and inside the vesicle we have the amino acids, and then the amino acid should be digested finally in the cell of the fungus. But here we have also the symbols with the ending SH. This is a cysteine. Among the different kinds of amino acids, keratin contains a high proportion of cysteine. And cysteine, this amino acid in the isolated form, is generally also toxic for cells. So when a fungal cell is taking up a high amount of cysteine, then it's intoxicating itself. And therefore, it needs to find a way to detoxify this cysteine. And therefore, there is some other particular enzyme inside the cell which is degrading the cysteine and is cutting off the sulfide from the cysteine. So then the cysteine does not exist anymore, but then we have sulfite, which is also toxic to the cell. So the problem is still not yet solved. And now the fungus is solving this problem by pushing out this sulfite. And so the sulfite perhaps may come in also by diffusion into the cell, but then there is a very effective transporter in the cell membrane, which is always pushing out the sulfite to the outside. And now we see there's another very great benefit, the sulfite, which is exported as a toxic molecule, is very helpful for assisting the enzyme which is breaking the disulfide bonds. So now the waste which is exported to the outside is assisting the fungus for getting food. So this is a very interesting and very great invention of the fungus for transforming the toxic waste into somewhat helpful or somewhat beneficial. So here we see the two steps of the toxic elements and then these toxins. There's a final toxic waste is exported and is assisting the fungus in getting its food. So that is just the same information, but in another way, in a more abstract way for illustration. So now we have the fungal cell with the blue circle on the right side, and the keratin is on the outside. So the fungus encounters the keratin and wants to degrade it, and then it's very difficult. So the fungus must invent new enzymes, the keratinases. This is the first new invention for the fungus. And then the keratinases crack down the keratin and release cysteine. Cysteine is imported into the cell, but then is intoxicating the fungal cell itself. So then there's no real benefit. So for dealing with this new problem, the fungus needs to decompose the cysteine by cutting off the sulfite. So then there's a new enzyme inside the cell. And then the cysteine is detoxified, but now a new toxin is coming up, the sulfite. And therefore the fungus is newly inventorying the sulfite transporter in the plasma membrane. So the sulfite is exported. And then this exported sulfite is helping the enzymes in degrading the keratins. So this is just only an example how complex interactions in nature and in the human bodies are. I think that is important to understand this principle with this single example, because as humans we always believe that we have found in one new medicament or new molecule or new enzyme, and then we are very happy about this new finding. But we should be very careful when we believe that we can solve a problem with our new finding, because nature and the human body are much more complex than we can imagine. And there are many other more complex interactions which may interfere with our new solution of a problem. So that was an example of the process and the mechanism of keratin degradation by fungi. So when we have a look on the fungi, we actually find that this ability to degrade keratin is very rare in fungi. But when we want to demonstrate that the fungus can degrade keratin is not so easy because there are not so many test methods developed. So there is basically there are two methods for routine detection of keratinolytic ability, and one is whether a fungus can or cannot degrade keratin directly, and usually we can use human hairs or hairs generally, and the other is with a pigment. So I will show to you these two methods. But principally, there is not yet a standardized method for proving keratinolytic ability. So here I show you photos from our own previous publications of human hair, which is inoculated artificially by a fungus, where we wanted to know whether it has or has not a keratinolytic ability. And you see here by microscopy, the blue colored parts is a fungus. The fungus is in blue and the rest is the hair. And when you see that in the microscope, it's very difficult to decide with a microscope whether the fungus is growing on the surface outside of the hair or whether it's penetrating into the hair and is growing inside. So this needs some skills in light microscopy. This cannot be simply done by a student or by a technician. You need some more experience in light microscopy for distinguishing clearly whether the fungus is growing just on the surface of the hair, which does not prove anything, or whether it's really penetrating into the hair and growing inside, which would indicate keratinolytic ability. And